For decades, endocrinologists searched for a singular metabolic master switch in the human brain. The massive, sustained weight loss achieved by modern incretin therapeutics like semaglutide is the result of that long search. Historically, the cornerstone of this entire field of study has been the melanocortin 4 receptor, or MC4R, pathway. In the classical, linear model, satiety begins when POMC neurons synthesize alpha-MSH. It must activate the MC4R bottleneck to trigger fullness. But if a patient has a mutated, non-functional MC4R gene, the signal stops, resulting in monogenic obesity and endless biological hunger. Under standard medical teaching, an intact functional MC4R bottleneck was considered the absolute non-negotiable prerequisite for any pharmacological weight loss. That rigid biological dogma collapsed during the recent SURMOUNT-1 trial, alongside isolated case studies of patients with homozygous pathogenic MC4R mutations. This chart compares weight loss percentages from those trials. Patients carrying completely broken pathogenic MC4R mutations still lost 18.3% of their body weight on modern GLP-1 and dual agonists, matching the nearly 20% loss seen in non-carriers. Under the classical textbook model, this data is biologically impossible. You cannot trigger satiety if the sole receptor for satiety is physically broken. The clinical efficacy of these drugs proves the brain lacks a single metabolic bottleneck. Instead, GLP-1 bypasses MC4R by executing a dual-mechanism rewiring of the hypothalamus. To understand how modern obesity drugs actually cause weight loss, we have to abandon the master switch theory entirely and map the molecular connectomics of this bypass at the single neuron level. We start deep within the hypothalamus, inside the arcuate nucleus. Its porous blood-brain barrier makes it uniquely accessible to circulating hormones and large pharmacological molecules. The first prong of GLP-1's bypass is a direct assault, targeting and actively exciting the anorexigenic POMC neurons. This diagram illustrates the membrane of a POMC neuron. When a GLP-1 molecule binds to the receptor, it couples to a G-alpha-S protein. This instantly triggers an intracellular cascade, elevating levels of cyclic AMP deep inside the cell. That cyclic AMP elevation activates protein kinase A, initiating a critical shift in the neuron's electrical excitability. Natural endogenous GLP-1 released after a meal creates a fleeting signal. Pharmacological GLP-1 receptor agonists are engineered to be relentless. The drugs achieve this durability by recruiting specific molecular hardware, TRPC5 subunits and L-type calcium channels, locking them into an open position. This continuous influx of positive ions creates a persistent membrane depolarization. The POMC neuron is forced to fire action potentials long after the initial drug binding event. Simultaneously, GLP-1 enhances presynaptic glutamatergic inputs. The entire local circuitry surrounds the POMC neuron with an inescapable excitatory tone. By hardwiring the brain's stop-eating signal to remain active, the drug ensures a powerful anorexigenic output. But activating satiety is only half the battle, and this direct excitation does not explain how the MC4R bottleneck is bypassed. Metabolic biology demands a balance between competing signals. Persistently activating a satiety circuit fails if the hunger drive is not simultaneously suppressed. The primary drivers of this intense hunger are the orexigenic AgRP neurons, located immediately adjacent to the POMC neurons in the arcuate nucleus. Here, researchers encountered a severe anatomical obstacle. AgRP neurons do not express any GLP-1 receptors. How can a pharmacological agonist silence a hunger neuron that it physically cannot bind to? This 3D schematic identifies a specialized interneuron known as TRH-ARC, recently mapped through molecular connectomics. TRH-ARC neurons have a dual identity. They express GLP-1 receptors to receive the drug's signal, but are strictly GABAergic, inhibitory cells. The mechanics are precise. GLP-1 activates TRH-ARC, which immediately fires a targeted barrage of inhibitory GABA neurotransmitters directly onto the AgRP cell. Operating alongside KATP channels, this localized GABAergic tone severely hyperpolarizes the AgRP neuron, suffocating the hunger signal at its point of origin. This explains the paradox of the monogenic obesity patients. By destroying the pathological hunger drive at its root, GLP-1 removes the behavioral necessity for the downstream MC4R satiety switch. It is a biological bypass. The effects of GLP-1 extend far beyond the localized arcuate nucleus, scaling up to macro circuits that connect the hypothalamus to the brainstem. A critical communication pathway in this network is the descending regulatory loop, originating from GLP-1 receptor-expressing neurons in the paraventricular nucleus, projecting down to the dorsal vagal complex. The synaptic strength of this specific pathway dictates how well the brain maintains a state of satiety and prevents overeating in a fed state. This diagram compares the integrity of that connection. Chronic overnutrition physically degrades the communication along this loop, creating a synaptic weakness that prevents the brain from recognizing a caloric surplus. Pharmacological GLP-1 receptor agonists provide the signal intensity required to override this structural weakness, forcing the descending circuit back online. GLP-1's clinical power relies heavily on this vast systemic redundancy. Addressing obesity requires repairing a distributed network rather than flipping a single receptor switch. Despite this distributed efficacy, one final hurdle remains: the significant individual variability in how patients respond to these therapies. Within the melanocortin system, this variability is regulated by the melanocortin 3 receptor, or MC3R. This conceptual panel illustrates that MC3R is not an on-off switch. It is a dial controlling energy rheostasis, an evolutionary upper limit that caps weight loss to protect the organism against starvation. Recent breakthroughs reveal that inhibiting MC3R causes the brain to become radically hypersensitive to GLP-1 drugs. Lowering this upper bound allows vastly smaller doses to achieve massive clinical weight loss without triggering the typical gastrointestinal side effects. Exploiting these interconnected limits represents the immediate future of polypharmacology in precision medicine. The clinical superiority of dual agonists like tirzepatide proves that hitting multiple network nodes simultaneously results in a profound suppression of the hunger drive. The evolution from hunting for a single master switch to mapping the brain's redundant plastic synaptic networks has provided the first clear blueprint for the long-term pharmacological management of metabolic disease.